Good morning, students. Today we are going to start a new chapter, that is biotechnology, principles and processes. So children, talking about the biotechnology actually deals with the technique of using live organisms or enzyme which obtained from organisms. Why? To produce products which are useful to human beings. For example, children, nowadays in vitro fertilization is very popular, which leading to a test tube baby, right? Developing of vaccines, correcting a defective genes. So these all are part of biotechnology. And in that way, biotechnology can be defined as the integration of natural science and organisms, cells for product and the services. Right? Children, talking about the biotechnology we can say of two types. The first one is the classical biotechnology and second one is the modern biotechnology. When we talk about the classical biotechnology, we can say that it is the use of living organisms since ages, like formation of organic acid, for example citric acid, and formation of ethyl alcohol, that is ethanol. तो ये formation use करते हैं living organisms और microorganisms. We already studied in previous chapters. And the fermentation, यहाँ भी living organisms ही use होते हैं, right? ये classical biotechnology है. We can also include curdling in the classical biotechnology, that is lactic acid bacillus, which is a bacteria, a living organism used for the human beings. So it is also included in the classical biotechnology. Now when we talk about the modern biotechnology, mainly it is related with the genetic manipulation, or we can say manipulation of DNA. DNA को मैनिपुलेट कर न्यू सेगमेंट ऑब्टेन किए जाते हैं ताकि न्यू प्रोडक्ट ऑब्टेन हो. Okay, so it includes recombinant DNA technology. So all this part comes with the modern biotechnology. Now when I say recombinant DNA technology means recombined, or we can call genetic engineering. So the core technique that enables birth of modern biotechnology are classified into two categories, that is genetic engineering and second is maintenance of sterile condition. As we know, the first aspect is the genetic engineering. So genetic engineering is the technique of altering the chemistry of DNA and RNA so that it can be introduced into the host organism to change the phenotype of the host organism, right? And the second aspect is the sterilization method. This method are used to maintain the growth and manipulation of desired microbe or cell. It requires to get large quantities of product, that is vaccines, enzymes, antibiotic, etc. In sterile conditions, it required for the chemical engineering processes, or in the simple word we can say maintenance of sterile ambience in chemical engineering process. Children, recombinant DNA technique के थ्रू जो नए रिकॉम्बिनेंट ऑर्गेनिज्म बनाए जाते हैं एंड इन ऑर्गेनिजम्स को एक माइक्रोबियल कंटेमिनेशन फ्री मीडियम में इन्हें ग्रो किया जा सके और इनके प्रोडक्ट को लार्ज स्केल में ऑब्टेन किया जा सके. तो ये दोनों एस्पेक्ट की वजह से रिकॉम्बिनेंट डीएनए टेक्नोलॉजी एंड स्टेरिलाइजेशन modern biotech has become possible, right? So because of these two techniques, modern biotechnology has become possible. So the important aspect of modern biotechnology, that is recombinant DNA technology. Okay, so recombinant DNA technology is the important aspect in modern biotechnology, right? So what is recombinant DNA technology? So how this recombinant DNA come in existence? Children, Stanley Cohen and Herbert Boyer produced first recombinant DNA by adding antibiotic resistant gene of Salmonella typhimurium in plasmid of E. coli. So this was first recombinant DNA produced by Cohen and Boyer. They produced recombinant DNA by integrating antibiotic resistant gene of Salmonella in plasmid of E. coli. So what is actually recombinant DNA technique? So in recombinant DNA technique, a specific DNA segment is isolated from one cell, and that particular DNA segment or gene is integrated into another specific cell, so that a new segment is obtained, and the new obtained DNA segment is called recombinant DNA, and this technique is known as DNA technology, recombinant DNA technology, or genetic engineering. Children, there are three basic steps of genetically modifying an organism. The first one is identification of DNA with the desirable gene. Second one is introduction of identified DNA into the host. And then maintenance of introduced DNA in the host and transfer of DNA to its progeny. So in that way, the technique recombination DNA technology occurs. So by those three steps, DNA technology, recombinant DNA technology occurs. For example, children, insulin synthesize करने की जीन होती है. ह्यूमन से बीटा पैनक्रियास के बीटा सेल्स से इंसुलिन सिंथेसाइज करने के जीन्स को आइसोलेट किया गया, ओके, और उस जीन को ई कोलाई के प्लाज्मिड में ऐड कर दिया गया, और जो नए ई कोलाई बने वो कैसा है, वो है recombinant plasmid, और टेक्निक इज कॉल्ड जेनेटिक इंजीनियरिंग, that is recombinant DNA technology. Okay, so when we talk about the technology, when we talk about the definition of recombinant DNA technology, so recombinant DNA technology is the process by which a DNA molecule can be isolated from a specific cell and can be inserted in another DNA molecule at desired position to obtain recombinant DNA. Okay, so this is about the recombinant DNA technology. What are the steps? Identification of DNA with desirable gene, introduction of identified DNA into host, and maintenance of introduced DNA in host and transfer of that DNA into their progenies. So this is about recombinant DNA technology. Now children, what are the requirements? Right, here we are going to talk about tools. Genetic engineering can be accomplished only with the uses of tools. What are those tools? The important tools of recombinant DNA technologies are restriction enzymes, cloning vector, competent host, and bioreactors. So these are the important tools required for the recombinant DNA technology. Children, when we talk about the restriction enzymes, so basically इन important tools की requirement होती है. इन चारों को यूज कर recombinant DNA produce किया जाता है, right? So talking about restriction enzymes are called a molecular scissor because these enzymes cut at the DNA at this specific site. The second important tool is the cloning vector. Like plasmids and bacteriophages have the ability to replicate within the bacterial cells independent of the control of chromosomal DNA. The third important tool is competent host. The host should be competent enough to take up the foreign DNA. So basically these three important tools are required. And another one is the bioreactor. So bioreactor is the cylindrical vessel in which biological processes is carried out on a large scale, right? So these are the different tools which are required for recombinant DNA technology. So we will discuss about each of them in little more detail one by one. So understood children, what is recombinant DNA technology? What happens in DNA technology? In recombinant DNA technology, the DNA fragment is taken from one cell, inserted into the vector, and finally recombinant DNA is there, right? So this is about the recombinant DNA technology. So first of all, let us look at the very first tool, that is enzymes. The restriction enzymes. So restriction enzyme belong to a larger class of enzyme that is called nucleases. Children, restriction enzymes are called as molecular scissor. Why? Because these enzymes cut the DNA at the specific site. The first restriction endonuclease is HindII2, right? The restriction enzyme cut DNA at specific base sequences, and these specific base sequences are known as recognition sequence. अच्छा, यहाँ पे restriction enzyme यूज होता है इस टेक्निक में. तो सबसे पहले हम बात करते हैं enzymes की. Children, let us talk about the enzyme and its categories. Right? So the very first category is the lysing enzyme, then restriction enzyme, then alkaline phosphate, DNA ligase, synthesizing enzyme and terminal transferase. अब यहाँ पे जो पहला एंजाइम मैं आपको बताने जा रही हूँ, that is lysing enzyme. ध्यान से सुनना. lysing enzyme में वर्ड यूज होता है lysis. lysis का मतलब होता है breakdown. In other words, these enzymes are used to open cell and separate DNA. जैसे कि लाइसो एंजाइम है, सेल्यूलेज है, पेक्टिनेज है, प्रोटीएजेज है. So they all enzymes are lysing enzymes and used to separate DNA. Got it? The second category is restriction enzyme. These are also known as molecular scissor, as I just mentioned, for cutting of DNA. So the cutting of DNA at a specific location become possible with the discovery of molecular scissor, that is restriction enzyme. Children, do you know who first isolate this enzyme? Yes, firstly Stewart Lynn and Arbor. These two scientists isolate enzyme which cut DNA and named it as restriction enzyme. Children, the first restriction endonuclease is isolated by Smith, Wilcox and Kelly. Restriction enzyme Stewart Lynn और Arbor ने isolate किए, और सबसे पहले restriction endonuclease isolate किए Smith, Wilcox और Kelly ने from the bacteria Haemophilus influenza. अब क्योंकि उन्हें हीमोफिलस इन्फ्लुएंजा से आइसोलेट किया था, इसलिए उन्हें नाम दिया गया HindII2. गॉट इट? तो पहला रेस्ट्रिक्शन एंजाइम HindII2. आगे पढ़ेंगे कि एंजाइम की नेमिंग कैसे होती है. जितने भी रेस्ट्रिक्शन एंजाइम होते हैं वो न्यूक्लियस कैटेगरी के होते हैं. अभी मैंने बताया, क्योंकि न्यूक्लिक एसिड को ब्रेक करके ये ऑब्टेन किए जाते हैं. सो दीज रेस्ट्रिक्शन एंजाइम बिलोंग टू द कैटेगरी ऑफ न्यूक्लियस एंजाइम which have 3 types. ये जो न्यूक्लियस एंजाइम होते हैं ये 3 तरीके के होते हैं. कौन कौन से? The first one is exonuclease, second one is endonuclease and third one is restriction endonuclease. Right? अब ये तीनों की कैटेगरी आपको समझाती हूँ. Exonuclease क्या होते हैं? What they does, they cut the terminal end of DNA. Let us suppose this is the DNA, and this exonuclease cut the DNA at the terminal end, right? And they are only capable to cut any end, not able to cut the intercalary segment. ये बीच में से नहीं segment को cut करते, ये एक terminal end से cut करते हैं, या तो इधर से या इधर से. ठीक है? The second one is endonuclease. What they does? They cut the segment ये क्या करते हैं? ये 2 कट करके सेगमेंट को रिमूव करते हैं. लेट अस सपोज दिस इज द सेगमेंट, दिस इज द डीएनए. एक कट यहाँ लगाते हैं और एक कट यहाँ लगाते हैं और ये सेगमेंट को अलग कर दिया. ठीक है. So they are capable to cutting intermediate or intercalary segment of DNA. Now the third one is restriction endonuclease. It is known as endonuclease, right? So why they are different from the second endonuclease? Endonuclease और restriction endonuclease में क्या फर्क है? Both above endonuclease or exonuclease cut any segment randomly, right? Endonuclease enzyme और exonuclease enzyme जो हमने डिस्कस करे वो रैंडम कट करते हैं, चाहे तो टर्मिनल एंड पर चाहे तो बीच में. And restriction endonuclease are the only endonuclease which are used in recombinant DNA technology. तभी यहाँ पे हम पढ़ रहे हैं restriction enzymes. क्यों? क्योंकि ये जो restriction endonuclease होते हैं ये specific enzymes होते हैं जो recombinant DNA technology में यूज़ किए जाते हैं. Why they are used in recombinant DNA technology? Because they identify the specific palindromic sequences और उन पैलिंड्रोमिक सीक्वेंस को कट करते हैं ये रेस्ट्रिक्शन एंडोन्यूक्लियस. ओके, सो रेस्ट्रिक्शन एंडोन्यूक्लियस दे रिकॉग्नाइज द स्पेसिफिक पैलिंड्रोमिक सीक्वेंसेस with the rotational symmetry and cleave the DNA within the sequences, right? So first let us talk about the convention of naming of the restriction enzyme, right? Then we will talk about the palindromic sequences. Let us see the convention of naming restriction enzyme. जैसे कि एग्जाम्पल हम इसी का लेते हैं, hint 2. The first letter is the name of genus. H is from Haemophilus. Second two letters are from the name of species, Influenza, IN. Fourth one is the strain, D strain, and the Roman number indicates the order of discovery, second discovery. So that is why hint 2. Similar example, Echo R1. So E from Escherichia, CO for coli, R is the strain and Roman number is 1, first discovery. So the name is Echo R1, right? So in that way the naming of restriction enzyme is to be done. Got it? So two categories of restriction enzyme we have studied, that is exonuclease and endonuclease. Exonuclease remove nucleotide from the end of DNA, whereas endonuclease make a specific cut and restriction endonuclease recognize a specific palindromic sequences, right? Now the palindrome in DNA is the sequence of base pair that reads same on the two strands when orientation of reading is kept the same. For example, children, in this example there are the directions 5' to 3' and 3' to 5', and the sequences GAATTC. अब इसकी mirror image क्या बनी, right? CTTAAG, right? So this is what? This is the palindromic sequence. Let us talk in little more detail. Palindromic sequence का मतलब like this is the sequence. ये एक strand है जिसकी सीक्वेंस है GAATTC और ये इसकी दूसरी कॉम्प्लीमेंट्री स्ट्रैंड है जिसकी सीक्वेंस है CTTAAG. ठीक है, इस तरीके के सीक्वेंस जो हैं वो पैलिंड्रोमिक सीक्वेंसेस कहलाते हैं. और पैलिंड्रोम्स में भी रोटेशनल सिमेट्री होती है. रोटेशनल सिमेट्री मीन्स इफ इट कट्स फ्रॉम मिडिल दे गिव्स मिरर इमेज बट इन अपोजिट डायरेक्शन. अगर हम इसे बीच में से कट करते हैं तो ये मिरर इमेज हमें देगा बट इन अपोजिट डायरेक्शन. जैसे GAA की मिरर इमेज AAG, CTT की मिरर इमेज TTC, बट इन द अपोजिट डायरेक्शन मिरर इमेज मिलती है. तो इस तरह की सीक्वेंसेस जो हैं वो पैलिंड्रोमिक सीक्वेंसेस विद रोटेशनल सिमेट्री कहलाती हैं, एंड ऑल्सो नोन एज पैलिंड्रोम विद रोटेशनल सिमेट्री. So restriction endonuclease only identify this type of palindromes. Now identify करके कैसे कट करेगा? उसके according restriction endonuclease further divided into 3 categories. The very first one is the type 1 restriction enzyme. कोई नाम नहीं दिया गया, बस type 1, type 2, type 3, right? तो type 1 क्या करते हैं? Type 1 identify the palindromes and also cut from any site randomly. Randomly वो कहीं से भी कट कर देते हैं. माना कि palindromic sequences हमारी GAATTC और CTTAAG. तो टाइप 1 क्या करेगा? टाइप 1 रेंडमली कट करेगा कहीं से भी, हेटेरोसेगमेंट्स दे फॉर्म, राइट? नाउ सेकेंड वन इज टाइप 3. इट ऑल्सो आइडेंटिफाई द पैलिंड्रोम बट ओनली कट फ्रॉम द साइड ऑन एनी पैलिंड्रोमिक सीक्वेंस. जैसे कि पैलिंड्रोमिक सीक्वेंस है GAATTC और CTTAAG, तो यहां से कट करेगा, यहां से कट करेगा. Whether homozygous segment is created, but it is not used in the recombinant DNA technology. Next category is type 2. Type 1 or type 3 are not being used in recombinant DNA technology, but type 2 is the only restriction enzyme which is used in recombinant DNA technology. क्यों? ऐसा क्यों? It always cut the palindromic sequences from the middle. ये जो है ये palindromic sequence को side से नहीं बल्कि middle से cut करता है. Only this type of restriction enzyme used in recombinant DNA technology because homogeneous segments produced. And children, do you know that over 900 restriction different type of type 2 restriction enzymes are known, right? And which are obtained from about 230 strains of bacteria. So these restriction enzyme identify specific palindromic DNA with rotational symmetry and cleave DNA with the specific sequence having similar nucleotide on the opposite side. Right? And more than 900 restriction enzymes have been isolated from 230 different strains of bacteria. Right? Now let us look at the mechanism of action of restriction enzyme. That now type 2 restriction enzyme cut the DNA segment. Type 2 restriction enzyme made two type of cut. First one is the blunt end cut and second one is the staggered or sticky end cut. When we talk about the blunt end cut, it is something like that. Like if the sequence is GAATTC CTTAAG, so the resultant segment are these. So these are the resultant segment of blunt end cut. इस तरह का कट blunt end cut कहलाता है. In this both the strands are cut at the same position. Second one is the staggered or sticky end cut. Suppose the sequence is the same and in this sequence the first strand cut from somewhere like here and the second strand is cut from here. So what are the sequences? This is the sequence 1 and this is the sequence 2. Now children, tell me which cut is perfect for recombinant DNA technology? The first one or the second one? Yes, the second one. क्योंकि पहले केस में अगर हम बात करते हैं तो दो सेगमेंट हमारे पास आते हैं, एक ये और एक ये. इन दोनों सीक्वेंसेस के बीच में कोई भी वेरिएबल लेंथ का डीएनए ऐड हो सकता है, राइट? बट व्हेन वी टॉक अबाउट द सेकंड सिनेरियो, सो इन दिस सेकंड सिनेरियो तो यहाँ पर जो बीच में जो डीएनए सेगमेंट ऐड होगा वो किसी भी वेरिएबल लेंथ का नहीं हो सकता, वो सिर्फ इन्हीं सीक्वेंसेस के बीच में एक परफेक्ट और स्पेसिफिक सेगमेंट होगा, राइट? समथिंग लाइक की एंड लॉक सिस्टम विच इज अप्लाइड हियर, राइट? तो यहाँ पर ब्लैंक है और यहाँ पर ब्लैंक है, यहाँ सीक्वेंस है और यहाँ सीक्वेंस है, तो कुछ ऐसा ही सिस्टम का इसी तरीके का सेगमेंट यहाँ ऐड हो सकता है। कोई भी किसी भी वेरिएबल लेंथ का डीएनए सेगमेंट यहाँ ऐड नहीं हो सकता. So that is why this sticky end cut is perfect for the recombinant DNA technology, right? And finally, these can be joined together by using the another enzyme, which is DNA ligase enzyme. DNA ligase enzyme is somewhat the molecular glue, right? They basically use to join the DNA segments, right? So children, I hope whatever we have discussed today, it becomes clear to you. In the next video, we will discuss some more concept of the biotechnology. So till then, have a nice day and thank you. And your task is to revise and learn today's topic. Thank you and have a nice day.